Conversations That Matter is a partner program for the Center for Dialogue at Simon Fraser University. The production of this program is made possible thanks to the support of the following and viewers like you.
Welcome. Thanks for joining me.
Great to be here, thank you.
We're at an interesting point in the mapping of the human genome. It's been about 25 years now.
Yep.
Take us back to the beginning. First of all, why did we map the human genome? And then let's work our way through to where we are now and what the promise of the future looks like.
Oh my goodness, how long do we have?
43 hours.
Well,
yes,
the Genome Project began in 1990, and if I cast my mind back, I was actually an active researcher in London at St Mary's Hospital Medical School, where I was doing my PhD. There were races to find the most important genes in the human genome, by which I mean from a human medical perspective, the disease genes, the genes that when mutated give rise to cystic fibrosis, muscular dystrophy, Huntington's disease, Alzheimer's disease, and then breast cancer and many other cancers. These quests involved and attracted the greatest minds in Canada, the United States, England, Europe, all over the world. And they took years, they sucked up millions of dollars in funding, in taxpayer funds, in resources, and great minds all competing to find one particular piece of genetic real estate. It wasn't efficient. It was
so
very
they were happening
wasteful.
in silos then, were they?
They were happening in silos, and we didn't have the tools to really go quickly. So it would take years to go from, first of all, pinpointing the rough location for a gene and then actually isolating it. So a good example is Huntington's disease, in which researchers at UBC, University of British Columbia, played a hugely important role.
Right.
That was a 10-year quest from 1983, saying, aha, we know that the gene is somewhere on chromosome number 4, to actually identifying the gene itself. 10 years of huge amounts of work and manpower hours. So the Human Genome Project was really sold to the United States Congress in particular as, let's build an information highway. Let's lay out once and for all the sequence of all of the genes across all of our chromosomes, from chromosome 1 to the X and the Y chromosomes, the sex chromosomes. And now when somebody is trying to identify the gene for either a rare disease or identify the genetic factors that contribute to the more common diseases that we all care about, that are much more complicated, multifactorial, type 2 diabetes, heart disease, cancers, we'll have the information there. It'll make our tasks so much easier. And when you go to Congress and you say, we've got something that could potentially help your constituents, who are all worried about Alzheimer's disease or prostate cancer, that sort of gets their attention. So they funded it as a $3 billion, 15-year project in the hands of a government coalition, an international consortium. And that's how the Genome Project was at least instigated way back in 1990.
Okay, we've done it, but what had we done, and what did that mean to the average person? What did it mean to our ability to actually do anything with that information?
Right.
And so here we are 15, 16 years later. What have we done with it?
Yeah, that's a great question. And the truth is, it didn't mean a whole lot to the average person. And I think probably because of the media hype and the way it was sold to Congress early on, that was probably a mistake on many people's parts, that somehow the promise of we've got the human genome. What it meant was, we have now sequenced the genetic units, the fundamental units of DNA, of our genetic code. So for the rest of our discussion, I'll call them letters or bases. Bases is the sort of the chemical shorthand notation, but think of them as letters, basically, the genetic alphabet. There's 4 letters, A, C, T, and G, which are the first initials of the 4 chemical bases. And if you think of the double helix, everyone's familiar with the double helix structure of DNA. It's like a twisted ladder. And the rungs of the ladder are made up of two of these letters that come together. They form pairs. A binds with T, C binds with G. So when you have the sequence of these letters on one strand, it automatically the opposite strand automatically kind of falls into place. And that's how our genetic code, our blueprint of life, gets replicated from cell to cell, to division, generation to generation. The Genome Project provided the first draft. It wasn't 100% pristine, but it was the first draft to a fairly high level of accuracy of what all 3 billion letters look like from the very tip? 3 billion.
Yep.
From the tip of chromosome 1 to all those 23 pairs of chromosomes that I mentioned earlier. And within that sequence of DNA, we know that there are now about 20,000 really important bits, fragments called genes. These are the building blocks. They typically code for proteins that make you and me, and everybody watching gives them their unique characteristics. And within many of those genes are genes that variations within cause genetic disease, determine risk to cancer, govern very much our ability to process different drugs. Hence this whole interest, very keenly held here in Canada about pharmaceutical research and pharmacogenomics, and learning how variations in specific genes in our body can influence our ability to process things like coffee. To drugs that we may take if we have a stroke, or cancer drugs, hugely important in terms of how, or statins, drugs that we may take to alleviate depression or schizophrenia. So this is a very fast emerging field. But I think when the Genome Project was finally completed, declared complete in 2000, and the scientific papers came out a few months later in early 2001, there was probably many people in the public who thought, well, that's it, right? Medicine becomes now a whole new era of designer drugs and cures for cancer. And frankly, the Genome Project itself was never going to deliver that, at least never, overnight. But it provides, I think that I'll end your question by saying it provides the foundation upon which now we can learn and begin to diagnose and treat cancer and mental illness and many of the other diseases that we care about.
I just got to get you to hang on for a second while we take a quick commercial break. We'll be right back.
Conversations That Matter is a not-for-profit program made possible thanks to the charitable support of the following and from viewers like you. Please visit conversationsthatmatter.tv and help us to continue to produce this program.
Shortly after, of course, the sequencing of the first genome, we start to get companies that are now saying, send us a sample of your saliva and we'll sequence your genome. And there was a tremendous amount of interest in that, but it wasn't full genome sequencing. And ultimately, what was the value of that information, especially in giving somebody some sense of, okay, what is my genetic code and what might I face as I age, which is a concern that so many people have.
Yes. Well, I can answer the first part of that question. The aging part let's perhaps come back to. But you're referring, of course, to companies like 23andMe, which was founded by the wife of Sergey Brin, the co-founder of co-founder of Google. There were several others as well doing a similar thing, where in 2007, when they launched, they invited consumers without medical supervision necessarily to gave them a DNA kit with which consumers could mail back a saliva sample, pretty painless, completely painless, from which DNA could be extracted, and the DNA is analyzed on something called a DNA chip. So this was not yet doing full-blown sequencing of your entire genetic code, but rather what 23andMe did is say, let's look at the 500,000 to 1 million specific letters in the human genome that we know govern our risk of different genetic diseases, different traits, ability to metabolize drugs. This is the real kind of the cutting edge of the human genome.
The genes of you as an individual.
Exactly.
Thus the 23.
23 pairs of chromosomes. So that's where the name of the company came from. And what 23andMe basically was and other companies like them were saying was, we don't need to sequence your 3 billion letters of DNA because there are some letters that aren't frankly that important. They don't change. They sit in kind of a gene desert that probably doesn't really matter a whole lot. But we're going to focus on not just the genes that we know are medically important or that are perhaps are useful in terms of determining your ancestry, but in particular we're going to look at the specific individual letters within that sequence, within the sequence of those genes that have been reproducibly shown to influence our ability to tell us something about health and disease. So this is what 23andMe did.
Okay, so when I look at what's happening in Canada right now with the Pharmacogenomics project, and it's being engineered through pharmacists, does that then start to meet that threshold that the FDA would require? And with looking at it from a pharmacogenomics perspective, me as an individual, do I start to say, yeah, I think I like that idea, because as you start to understand more about drug interactions, that's what I want to know. The disease part is one thing, but whether I'm going to get that disease or not, I still got to take drugs.
I think one area where perhaps the pharmacy community can really play a part is more in public education for how your genetics, your personal genome, can influence one's ability to metabolize drugs. If a doctor prescribes an antibiotic or something else for me, a triglyceride medication, whatever it might be, my first thought isn't, oh my God, how am I going to metabolize that drug? I don't really think about it. I mean, the doctor's written me a prescription, I fill it out, and I go home and take it. Well, they don't know. They don't have a clue. And I think...
And that's the problem, isn't it? Yeah.
And but for people in other areas suffering from other disorders, bipolar disease being, I think, an excellent case in point, one hears, and I know from some family members, and one hears anecdotally that, the challenge with treating people who suffer from depression or bipolar disease isn't necessarily finding the right drug, it's finding the right dosage of the right drug, because among all of the variations in our DNA, many occur in a series of different enzymes that that control our ability to metabolize drugs. So as we take a drug, whether it be for heart disease or a cancer drug or a drug to treat some form of mental illness, or caffeine in our coffee cup, we're all metabolizing these foreign molecules to different extents. Some metabolize them very fast and the drug is gone. Some very slowly or not at all, hardly at all. So it behooves us as consumers, as patients, and the medical establishment and the pharmacists that you mentioned to do a better job of revealing what these variations are and educating the medical community how this information can be important to guiding what you prescribe and in what dose you prescribe it. And I think one has to really tip one's hat to the work that they've been doing here in British Columbia at the UBC, this pilot program where they've identified specific stories of individuals who've been over-medicated, really having challenges because it's been a trial and error. But if you can now say a patient can go to their pharmacist or their prescribing doctor and say, here are the key variations in the genes that I metabolize the drug you're about to give me. I know I'm a slow metabolizer or a fast metabolizer. That gives the doctor precious information now to tailor the dose and make a much more rational, educated decision as to how to treat that person's disease, rather than just throwing drugs at them and hoping for the best.
Unfortunately, we've got to take a second break here. We'll be back in a moment.
Conversations That Matter is a not-for-profit program made possible thanks to the charitable support of the following and from viewers like you. Please visit conversationsthatmatter.tv and help us to continue to produce this program.
Well, one of the other things that I think about as you're explaining all of this is, okay, well, who collects that data, who houses it, who does that ongoing research, who has the key so that it's not held by one private company that says, oh, well, we're going to use this just for our benefit, forget all the other drugs. And so that becomes central in this too. Do we trust the organization that is going to be managing all of this information?
I think a lot of people are worried about genetic privacy and somehow the genetic information getting out somehow into the public domain. And if you don't have sufficient legislation, then yes, perhaps an insurance company, somebody governing your private health insurance or life insurance, that potentially could be misused. But I think perhaps a little bit more has been made of this than need be. I don't think people are going to be scrutinizing my genome too closely. I think I've heard people say that they're much more worried about their financial information being hacked than their genetic data.
Yeah, no, I was thinking about it from the perspective of who controls that information so that it's available to all. So everybody who's doing research around drugs, drug interactions, who is able to consume, understand, and then put that information to use. It's as though it has to be in a non-biased environment or entity.
Perhaps. I'm sure most people would be happy if the government or a non-profit set up that sort of entity. Although you brought up 23andMe earlier in our conversation, and one of the virtues of that privately held company was that they now have more than 1 million people in their database. What's attracted many of those people is the altruistic notion that they, by submitting their saliva and becoming an N of 1 in this N of 1 million database, they're helping the research enterprise. Their anonymity isn't being sacrificed, but piece by piece, those genomes are accumulating to the degree that is helping basic research. So over the last few years, 23andMe have published a number of papers in exceptionally prestigious journals using that crowdsourced data to identify new genetic risk factors for crucial, important diseases like Parkinson's disease, asthma. So we can pat ourselves on the back and say, our DNA was only a tiny infinitesimal fraction of that study, but we did our part to help these guys. Where I think some people resist that notion is if that data is then being licensed to a pharmaceutical company, which is then going to profit off it.
But it doesn't appear that the people who are working in this area, that's their motivation. It really is for the collective good.
It is for the collective good, although I will say that I think 23andMe's business model is evolving of late. They are showing signs of becoming much more of a drug development company, not only by partnering with some big pharma companies, but bringing in some very experienced pharma executives themselves, because they're sitting on this database of a million genomes. And you can do lots of fun recreational stuff, and they can tell me how much Neanderthal DNA I've got buried and lurking in
0.4%.
It's a little over 2% actually.
A little over 2%.
Yeah, sure.
For anybody whose ancestors came from north of the Sahara.
Something like that.
Yeah,
yes.
Okay.
Yes, a little lower for some others.
Yeah.
So there's that sort of fun recreational aspect of it. But if that information can give me insights into what are the genetic factors that control heart disease or asthma or autoimmune disease or celiac disease, then as a drug company, I've got to find ways to mine that data. And 23andMe, of course, it behooves them as a business to try to find ways to do that without compromising anybody's individual privacy or identity.
Last break, I always hate it, but have to do it. We'll be right back.
Conversations That Matter is a not-for-profit program made possible thanks to the charitable support of the following and from viewers like you. Please visit conversationsthatmatter.tv and help us to continue to produce this program.
So, we've been talking only about the human genome.
Yes.
We've also been mapping genomes of virtually everything else.
Yeah.
What is, now as we sit here in 2016, what is the promise of genomics as we move forward?
Well, I think
in all areas.
Yeah. You've got there's a huge interest in terms of sequencing plants and agricultural crops. Feeding the planet is going to be a hugely important challenge, and we get into how can we either breed or produce new strains, whether it's through natural crosses or using gene editing techniques if necessary. This gets into the whole genetically modified food debate, but it seems that there's really little option. If we're to feed 7 billion and counting people on this planet, we've got to have crops that can sustain drought and floods and cyclones and typhoons and all the rest of it. And we've got to increase the nutritional value of these things. So by sequencing the genomes of these different crops and different strains of rice and many other crops, that gives us hope that we can build a more sustainable planet for our future. Obviously there's work sequencing a whole bunch of different organisms. We're into some pretty esoteric things now. The sea walrus, the cucumber. We've sequenced a lot of very important organisms in the past. There's talk about the woolly mammoth genome and potentially recreating that. But there's still, if we're giving the impression that the work on the human genome is done, it's not. There's still, as the price of sequencing the human genome comes down, we're now we currently, as we sit here today, probably sequenced about 250,000 human genomes in their entirety.
Globally.
Yep. But let's push it on. How about 1 million? What can we learn from 1 million or 10 million? What if we do reach a point in the next 10 or 20 years where people are carrying some form of their human genome on their smartphone? What can we use that for? And I think there's still so much about human biology that we don't understand. So while we focus a lot here about talking about medical and pharmaceutical applications of the human genome, we still are trying to understand how the human body works. And you think of, for example, one of the offshoots of the Human Genome Project, the cancer moonshot that Vice President Joe Biden is leading. Everybody has been applauding this. It's the successor to President Nixon's war on cancer. But think of every cancer patient story that you've heard or read, or a patient, friend, family that you've known. Think of the heartache as they've tried one drug. It's looked promising for a while, and then they've relapsed, and they've taken other drugs and so on. I think we all can think of cases of our acquaintance where that's happened. But understanding how the genetic pathways that govern cancer, that give cancer the ability to resist it. When we hit it this way, there's a mutation that it allows it to escape this particular drug that we're giving it. So then we come at it from this side, and then it goes off in another direction. So there's still a huge amount more work that we have to do.
But we're at a pretty exciting point. We're starting to see it creep into our lives and have an effect on the way that we're living, being treated, and getting better.
Well, we are. I ended my book, The $1,000 Genome, talking about some of the new sequencing technologies that are on the horizon. There's one from the UK, from a company called Oxford Nanopore, which is in 2017 about to unveil a little device that will it'll be a DNA sequencing widget that plugs into your smartphone, just like a credit card reader, and you could load in DNA, and the material, the data would stream directly into your phone. I mean, let's see if it actually works, but based on their track record, I'm sure it will. So the cost of sequencing, the portability of sequencing, the fact I can now take this device or a device similar to it, I can take it to Liberia or Sierra Leone or some country in Africa that is reeling from an Ebola outbreak or a Zika virus outbreak, and actually now I don't need to ship samples to America or ship patients to America to be sequenced, quarantined and sequenced, or Canada. I can now take the lab to the outbreak and potentially learn what's causing the outbreak, learn where the key strains are, learn how it's evolving, where it's mutating, and treat the right people at the right time far earlier than we could before. That's amazingly exciting.
And that's just the beginning of the promise. Thank you very much.
Thank you so
Really
much. Thank you for having me.
Yeah.